Yeah, hi everyone. I'm Daniel. So I'm a postdoc in Abe's lab and also in Pejman Mohammadi's lab at Scripps Research. And so I'm going to show you our work on mapping and analyzing eQTLs in multiple brain regions of heterogeneous stock rats. And so as you all know, as Abe was talking about, rats are an important organism for mapping the genetic associations with different behavioral and physiological traits. But often with these GWAS loci, we don't know what the biological basis of them is, if there are any causal genes involved. And so the big motivation for this study is to map the genotype expression associations called expression QTLs and across the rat genome. And so this will allow for different things like identifying regulatory elements and being able to co-localize these with the GWAS QTLs, and that can identify putative causal genes. And in addition to helping understand the biology of it, involving genes can increase the relevance of these results to human biology. And so to do this, we obtained RNA sequencing data from 5 different regions of the rat brain from 88 heterogeneous stock rats. And so Abe had also talked about what the HS rat population is like, but this mosaic of different founder haplotypes provides us with a lot of genetic variation, which we obtained the genotypes from genotype by sequencing, and we quantified the transcriptomes from the samples from these 5 brain regions. And then this is showing what the haplotype level diversity looks like. So using the genotypes, we estimated the portions of each animal's genome coming from the founder strains. And we're kind of surprised to see that this was less uniform than we expected. But after we did some simulations, we found that this really falls in line with what we would expect from the amount of genetic drift you would get over time. But overall, looking at the genotype level, this leaves us with a lot of genetic diversity. If you look at the minor allele frequencies, they're fairly uniform and overall a lot higher than you would get with some populations like humans, and that gives us a lot of statistical power. So using this data, we mapped expression QTLs. So we modeled this loosely off of the GTEx pipeline. So this is the genotype tissue expression project that was conducted over dozens of human tissues over the last decade or so. And so we used single SNP linear regression using TensorQTL, and we mapped associations between every variant and genes expression levels. So you can see that the vast majority of the strong associations occur with variants close to the gene. And these are, we call these cis eQTLs because they're putatively involving the cis regulatory associations. And so we focused our analysis on these cis eQTLs that are within a megabase of the gene's transcription start site. So overall, over these 5 brain regions, we identified cis eQTLs in almost 8,000 genes. And using stepwise regression, we were able to identify multiple independent eQTLs for some of these. But this just gives you an idea of how much those eQTLs overlap across tissues and how many of them are tissue specific. And then the distance from the eSNPs to the transcription start site, you can see they're concentrated closer to the TSS within this cis window, which is consistent with just where enhancers and promoters tend to be located. And we validated these EQTLs using the orthogonal model of measuring the association. So instead of just the number of alternative alleles and the total expression, we can also look at the heterozygous individuals and measure the allele-specific expression for that SNP. And using these two different measures of effect size for the EQTLs, we confirmed in each brain region that the effect size is correlated pretty well, as we would expect. We also wanted to test whether it would be necessary to use linear mixed models in this context of cis EQTLs in HS rats. And so this is a way of adding a random effect term that models the genetic relatedness between the individuals. But overall, we found there's actually very little effect on the p-values of the cis EQTLs when we added that term. Another way of showing this is that whatever p-value the cutoff you have, you get almost the same results, the same set of EQTLs. And so I think this has a lot to do with the fact that there are some closely related individuals within this cohort of 88 rats, but overall, we don't see major clustering and population structure, which is a big motivation for linear mixed models. So for this reason, we just went with the GTEx approach of using a fixed effects linear model for our main results. And so finally, because we expect these EQTLs to be important, be useful for the rat genetics and genomics communities, we want to make it available. So we've initiated this Rat GTEx portal, which is available at ratgtex.org to host the data. So this is inspired by and with code adapted from the GTEx portal, which is for human tissues. But we are using a uniform data processing pipeline. And so that when we, on an ongoing basis, we'll add data sets from different tissues from additional studies, but we'll be able to compare results across the tissues and studies. So this portal provides some interactive data visualizations, such as the one shown here, and as well as data downloads and an API for the data, which, as Rob was talking about, will follow the FAIR principles using the Open AI framework. And here's another visualization that it shows. So there's a screenshot of one showing all of the EQTL results for a gene. And so you can see it shows all of the tissues, has different ways to filter. You can search by gene. So I think it has a lot of cool features, if I may say so. So to conclude, I've shown how we have mapped and analyzed EQTLs in 5 brain regions in heterogeneous stock rats. And that the EQTL discovery, both the methodology and the results, resemble what has previously been shown in humans, but with some important distinctions, such as the relative importance of population structure versus the importance of LD structure in the genome. And we have built the Rat GTEx data portal and have these these 5 tissues added to it to provide these and additional projects to the rat genetics and genomics research communities. So I'd like to thank the other members of the Palmer Lab and the PEDGE Labs for their support and ideas, as well as to the rat genomics and genetics communities, which we're glad to be a part of. And then this work was funded and organized by the NIDA Center for GWAS in Outbred Rats. So thank you, everyone, and I can take any questions now.